Chapter 6, I would say, is one of the most difficult chapters, is one of the most difficult process in biology and that you're going to have this semester, just because there are various steps to it, and each step or each stage, you have to understand what's going on, and there's things that you have to memorize. You know, there's this worksheet that is posted on Blackboard. Is in the Chapter 6 lessons folder in the worksheet assessments. There's one that is blank, and then there's one that has all the answers. So you can choose to do this worksheet as you're watching the video, or watch the video and then fill in the worksheet. So of course, you have a description of the process, and here are the 4 steps to the cellular respiration. So for glycolysis, a short, easy description is splitting of a sugar molecule, splitting of a glucose molecule. And then you'll have to know if each stage is aerobic or anaerobic. So anaerobic means that it doesn't need oxygen to proceed. And then aerobic means that it does need oxygen, with oxygen to proceed. And then you'll have to know where exactly in the cell or the mitochondria each stage takes place, what the reactants are. That means what types of molecules are going into the process, and then how many molecules it is, and then next, the products. That means that you have to know the types of molecules that are made from each process, and what the number of those molecules are. So that's a lot to remember. Okay, so let's have a summary of cellular respiration. We've actually talked about cellular respiration a few times before. The purpose of this process, this entire process, is to make ATP, which is a chemical form of energy that our cells will use to sustain life, to do whatever functions, to carry out whatever functions it needs to do. And then here is the chemical equation for cellular respiration. This is an equation that you should remember, okay? So here we have 1 molecule of glucose, okay, that we intake from food. And then we take that and add it with 6 molecules of oxygen, okay? So 6O2, that's the air that we breathe in. We combine those two, and then we make our ATP. So a byproduct of cellular respiration that we produce are 6 molecules of carbon dioxide. And then also in the process, we make 6 molecules of water. 6 molecules of water. So here we have our reactants, the molecules that go into the process. And then here we have all the products. So these percentages here, from 1 molecule of glucose, we only extract 34% of that potential energy from glucose in the form of ATP, and then the rest of that potential energy from glucose goes off in the form of heat, an unusable form of energy. You may think, oh, we're wasting 66% of potential energy and we're only extracting 34%. Actually, this 34%, we actually produce a lot of ATP, about 32 molecules of ATP per 1 molecule of glucose. So that's actually a lot of ATP. Okay. And then now let's talk about the important players of cellular respiration. We have these molecules called coenzymes, and then these coenzymes are organic non-protein helper molecules. What does that mean? Well, they shuttle things from one place to another place within the cell, within the mitochondria. So you can think of them as carrier molecules. They can either carry electrons, hydrogen ions, and then another word for hydrogen ions are protons, or they can also shuttle chemical groups. And then these coenzymes we can find in our vitamins. So this is why it's so important to extract vitamins from our foods. And then if we don't, if it's not in our diet, then we can take supplemental vitamins so that we can carry out cellular respiration. What these coenzymes do is that they just help the enzyme for the substrate to fit better into this active site. And then there's 3 different coenzymes that are key players in cellular respiration. The first one is called nicotinamide adenine dinucleotide, and abbreviation for that is NAD+. NAD+. And then these are your niacin or vitamin B3, and these are some of the foods that you can find, niacin in. Another one is flavin adenine dinucleotide, or FAD. FAD. So these 2 coenzymes, they have a very special function. They actually shuttle electrons around and hydrogen ions around. And then your last coenzyme is just called coenzyme A, and the abbreviation is CoA. This one actually does a little bit of a different function. It shuttles chemical groups from one place to another place. Okay, so now I just want to give you some background information. So the whole point of cellular respiration, how we get that energy, is through the bonds, through this hydrogen ion, through the hydrogen atoms. So this is the chemical structure of one glucose molecule, and you can see that there's a lot of hydrogen atoms on them. Okay, and then what we do is that we break those bonds up, and then we extract the hydrogen ion and then the electrons from the hydrogen atoms, and then those could be used as energy. Those represent potential energy. And then here we can see that our hydrogen atom, it consists of 1 proton and then 1 electron. So it would be the same as here we have 1 hydrogen atom, and then if we break it down, it would be 1 proton or 1 hydrogen ion and then 1 electron. So these 2 are what those coenzymes NAD plus and then NAD will potentially become NADH and FADH2, shuttle these protons and electrons around. And then these are the equations to represent how NADH and FADH2 are made. So here we start off with our NAD plus. It accepts 2 electrons along with 1 hydrogen ion, and then it'll become NADH. NADH. So here, this NADH, it will shuttle 2 electrons around and 1 proton. Here we have FAD, and then it'll combine with 2 electrons, 2 hydrogen ions to become FADH2. So this coenzyme, this FADH2, it'll shuttle 2 electrons around and 2 protons around from one place to another place. Here, your coenzyme A, it will actually combine with a 2-carbon group called an acetyl group to become acetyl coenzyme A. So this coenzyme, what it's doing is that it's shuttling or bringing this acetyl group from one place to another place. So this picture actually found on the internet, I think is really great. It kind of just summarizes what I just said in a more realistic or a real life example. So here you can think of these NAD plus and FAD as a taxi, an Uber or Lyft. And then here the passengers are electrons or even hydrogen ions Okay. So the purpose of a taxi is to pick up passengers, take them from one destination to another destination. So now this loaded taxi, it has those two electrons, it has the one proton for NAD, and then it has two protons for FAD. So now it's become NADH, FADH2 when it's loaded, when the taxi is full. So now it gets to its destination, and then it empties the passengers. So it empties the electrons and the protons. It drops them off. So after they've dropped the electrons and the protons off, it goes back to its original form of NAD plus and FAD. And then what this taxi is going to do next is that it's going to pick up some more electrons, some more protons, and keep shuttling them around. The first three steps of cellular respiration is really not to make ATP. You do make a little bit of ATP, but the main purpose of the first three steps is to make as much NADH and FADH2 as you possibly can, because these electrons, these protons, they represent potential energy. And then, in laws of thermodynamics, you can't create energy, you can only transform them. So we're taking this potential energy and we're going to transform it into ATP energy. And then here you have your acetyl coenzyme A. So here's that coenzyme A, and it's going to pick up an acetyl group, which is a 2-carbon molecule, and then shuttle it to destination and then drop those off. And then it'll go off and pick up some more acetyl groups. So keep these players in the back of your mind while we're talking about cellular respiration. So that was the first 3 steps of cellular respiration. And then the very last step is when you have made as much NADH and FADH2 as you possibly can. And then you go to the last step of cellular respiration, and then you drop off those electrons, you drop off those protons from NADH, and also FADH2, you drop off those electrons, you drop off those protons, okay? And then it'll go through a series of electron carriers, and then that energy will be used to make ATP. So you have these electrons, you drop them off, but where do they go? You can't just have electrons floating around in your cell. That's potential energy. You have to have these electrons bound to something to neutralize it. So that's where oxygen will come into play. So oxygen will bond to electrons and protons to make water. So in cellular respiration, oxygen is your final electron acceptor, and then you'll produce water. Okay, so now we have to know about the structure and the anatomy of the mitochondria, the site of cellular respiration. So remember how I told you that the mitochondria is a very, very important organelle. Well, since it's so important, it needs to have 2 membranes. So we have an outer mitochondrial membrane, okay? That's a phospholipid bilayer. And we also have an inner mitochondrial membrane. That's also another phospholipid bilayer. And then what happens with this inner membrane is that it kind of has folds. And then the reason why it has folds is to increase the surface area. Okay, so anything in your body that has folds is to increase the surface area. And then these folds, they're called cristae. And then this inside compartment is called the matrix, the mitochondrial matrix. And then on the inside of the mitochondrial matrix, you have structures. You have DNA, you have ribosomes, and also you have enzymes. Okay, and then these enzymes are very important because these are the enzymes that will carry each step of the each step of the chemical reactions of cellular respiration. So keep these structures in mind as we're going through the process, each step of cellular respiration. Okay. So for cellular respiration for this chapter, you'll need to know where everything happens, if each step is aerobic or anaerobic, what the reactants are, what the products are, okay? So let me give you a brief summary of cellular respiration. So the first step is called glycolysis, and then glycolysis will actually occur on the outside of the mitochondria. So you can say it occurs in the cytosol or the cytoplasm. And then we take one molecule of glucose, we break it up, and then we produce something that's called pyruvate. We actually produce 2 pyruvates. And then in the process, we make 2 ATPs, okay? And then in glycolysis is anaerobic. We don't need oxygen yet. So we go through glycolysis without oxygen. It's an anaerobic step. And then also in the process, you make 2 NADH. Good, because that's the purpose of glycolysis. That's the purpose of the first 3 steps of cellular respiration. So each of these NADHs, they'll carry 2 electrons and 1 proton. And then now if you have oxygen, you can go into the mitochondria. If you don't have oxygen, you stay in the cytoplasm. So let's pretend that we have oxygen. So all these steps down the line will be aerobic. So that means that you need oxygen for these steps to proceed. So the second step is called pyruvate oxidation, but we'll call it formation of acetyl coenzyme A. So in this step, you produce 2 acetyl coenzyme A, and then you also produce NADH and carbon dioxide. So good, we've produced NADH. And then the next step, we go and produce, we go into what's called the citric acid cycle. And then another name for it is Krebs cycle. And then in this process, we actually make a lot of NADH, and we finally make some FADH2. And then we make a little bit of ATP. So these 2 steps will occur in the mitochondrial matrix. And then the last step, the 4th step, we will go into the inner mitochondrial membrane where electron transport and chemiosmosis will take place. And then collectively, they're called oxidative phosphorylation, but you don't have to worry about knowing that it's called oxidative phosphorylation. Just know it as electron transport chain and chemiosmosis. And then it's in this process where we're going to take all the NADHs and then the FADH2s that we've made, drop off the electrons, drop off the protons, so that those can be used to make a lot of ATP. And then those electrons and protons, they'll combine with oxygen to make water in this final step. So here, your book, it says that we make about 32 ATP. So you really never know exactly how much ATP you make per molecule of glucose, just because there's so many things going on. Not all the NADHs will be used to make ATP. Not all of the FADH2s will be used to make ATP. So this number, the number of ATPs, we will never ask you, what is the final result? What is the final number of ATP? Because some sources will say 32, some even will say 30, 34, and even 38. So that number is really kind of unknown. But these numbers here, 2 ATP here, 2 ATP, those are known. Okay. So please excuse my handwriting. So this is what I would usually write on the board, but this is an online class, so I wrote it more neatly for you guys. So here, the first step is glycolysis, and glycolysis occurs on the outside of the mitochondria, but within the cell. So it's in the cytosol or the cytoplasm. And then remember that we don't need oxygen to go through glycolysis. Okay, so it's an anaerobic step. So what happens in glycolysis is that glyco means sugar, lysis means to break or split. We start off with one molecule of glucose, and then this glucose is a 6-carbon sugar. So 1, 2, 3, 4, 5, 6. Okay, so what we do is that we break the 6-carbon sugar glucose in half, and what we produce is a 3-carbon molecule called pyruvate. So here's one pyruvate that you've made, and here's another pyruvate that you've made. But for us to even get the whole process started, we need to put in some energy. So here we use 2 ATPs to get the chemical process get going. And then in the process, we've made 4 ATPs. So that means we have a net of 2 ATPs, a net of 2 ATPs. So remember, do you guys know that saying, you got to put in money to make money? So here we're putting in money or putting in energy, and eventually we're going to make a lot of money or a lot of energy. But the point of this process, glycolysis, is not to make the ATP, it's actually to make NADH. So we will be making 2 NADHs in glycolysis. So what happens is that you have 2 NAD+, and each of them are going to pick up 2 electrons and 1 proton, and then the other protons will go off into the medium, into the cytosol. So here we've made 2 NADHs. Great, because that's the purpose of glycolysis, to gather up as much energy as possible, to gather up as much NADHs as possible. So now we have the second step, which is called formation of acetyl coenzyme A. So this step, you do need oxygen, okay? And then when you have oxygen, you are allowed to go into the mitochondria. And then this second step specifically happens in the mitochondrial matrix. So that inside space, inside of that inner membrane, where you have the ribosomes, enzymes, and DNA. Okay, so from glycolysis, we made 2 pyruvates. And then now the first step of the second step, the formation of acetyl coenzyme A, is what you're going to do is that you are going to cleave off or break off one of the carbons. And then that carbon will go off as carbon dioxide. It'll go off as carbon dioxide. So what you do to one pyruvate, you have to do to the other pyruvate. So this carbon will break off or be cleaved off, and it'll go off as carbon dioxide. And then now you're left with a 2-carbon group called an acetyl group. And then this acetyl group will be picked up by coenzyme A. Coenzyme A. So here, this acetyl group is also picked up by coenzyme A. And then so for each pyruvate, so for this pyruvate, you've made 1 carbon dioxide. Okay, so that's the carbon dioxide that you exhale out, that you breathe out. So this is the first step that you have produced carbon dioxide. That's a distinctive step. And then in the process, the purpose of the second step is that you're making your NADHs. So from 1 pyruvate, you've made 1 NADH. Okay, so that's from this pyruvate. And then the same will go for this pyruvate. You've made 1 NADH, 1 carbon dioxide, 1 acetyl coenzyme A. But total, you've made 2 NADHs, 2 carbon dioxide. So we can say from 1 molecule of glucose, we've made 2 NADHs, 2 carbon dioxide in this step, in the formation of acetyl coenzyme A. From 1 molecule of from 1 molecule of pyruvate, we've made 1 NADH, 1 carbon dioxide, 1 acetyl coenzyme A. So you'll have to know how many molecules of what we've made from 1 molecule of glucose. You'll have to know, what molecule and the number of molecules we made from 1 pyruvate. This third step it has multiple names. Most of the time we'll refer to it as Krebs cycle, and then sometimes it's called the citric acid cycle. And then also is called the tricarboxylic acid cycle, or the TCA cycle. So don't worry about that one. That's a really long one. This one, we are still in the mitochondria. We still have oxygen. So this step is aerobic, has oxygen. And then it occurs in the mitochondrial matrix. So here, from the formation of acetyl coenzyme A, we have 2 acetyl coenzyme A molecules, okay? And then each of these molecules, what's going to happen is that they're going to go into a cycle, okay? So before they can go into the Krebs cycle, what needs to happen is that this coenzyme A, the taxi, is going to drop off the 2-carbon acetyl group. So this actually will go into the citric acid cycle or Krebs cycle. It'll activate the cycle. So your coenzyme A doesn't actually go into the cycle. It'll just drop off your acetyl group. And then what's going to happen is that this coenzyme A is going to pick up some more acetyl groups to drop them off into another cycle. So since we have 2 acetyl groups, we will need 2 turns of the Krebs cycle. Let me say that again. Since we have 2 acetyl groups, we will need 2 turns of the Krebs cycle. And each turn of the Krebs cycle will produce 3 NADH, 1 FADH2. So this is distinctive. This is the first step and only step that we've actually made our FADH2, the other type of coenzyme A, the other type of coenzyme, sorry. And then here, we've also made 1 ATP per acetyl, 2 carbon dioxides. So whatever happens to this acetyl group needs to happen to this acetyl group. So each turn of this Krebs cycle will produce 3 NADH, 1 FADH2, 1 ATP, and 2 carbon dioxides. So total, if you add up both of those turns, you get a total of 6 NADH, 2 FADH2, 2 ATP, and 4 carbon dioxides. So the step that produces the most carbon dioxides that you exhale out is the Krebs cycle. And then you can say, for example, for 1 molecule of glucose, we've produced 6 NADH, 2 FADH2, 2 ATP, 4 carbon dioxide in this step of the Krebs cycle. But per molecule of acetyl group, we produce this. And you can also say per turn of the cycle, we've produced 3 NADH, 1 FADH2, etc., etc. So it's important that you look at it from those perspectives. And then when you see this book, if you're trying to read the textbook to gather some more information, don't worry about the intermediates like citrate, alpha ketoglutarate. You don't need to memorize that for the exam, but feel free to, of course, read some more. But if you count it up, there's 3 NADHs that have been produced, and each of them will carry 2 electrons and a proton each. And then here, this Krebs cycle is finally when you produce your FADH2s. And each of them will carry 2 electrons and 2 protons. Okay, so those 3 steps, it had one purpose. It was needed to produce as much NADH and FADH2s as possible to carry those electrons, to carry those protons from those steps into the final step, the 4th step, which is called the electron transport chain and chemiosmosis. And then this will happen in the inner mitochondrial membrane. And then, of course, since we're in the mitochondria, we'll have oxygen, so it is aerobic. And it's here, this final step, where we will make a lot of ATP, So total from all 3 steps, we've made 4 ATPs, so a little bit of ATP. We've made 6 molecules of carbon dioxide, and then 10 NADHs. So if you add up all those NADHs, it'll add up to 10. And then 2 FADH2s from that Krebs cycle, and then we'll take our oxygen, and then we'll make a lot of ATP. And then if you put it all together, it'll look somewhat like this, but probably neater. Okay, so you have your glycolysis. So this is the only step where it'll be anaerobic, okay, where it won't have oxygen. And then if you do have oxygen, you'll proceed to go into the mitochondria to proceed to steps 2 to 4. Okay. So the last step, step 4, is pretty complicated just because there's so many things going on. Here we have something that's called the electron transport chain. And then this electron transport chain is just a series of proteins. Okay, so here's a protein, protein, protein. Here's protein as well. And then what they do is that they will shuttle electrons around from one place to another. And then when they shuttle the electrons, it'll energize these proteins to pump hydrogen ions from one part of the mitochondria to another part. Okay. And then chemiosmosis, what you're going to do is that you're going to build up a high concentration gradient of hydrogen ions. And then this represents potential energy. And then what's going to happen is that that high concentration of hydrogen ions is going to energize this enzyme called ATP synthase to make a lot of ATP. And then that's called chemiosmosis. So that's the gist of this picture. So let's try to break down this picture in more detail. So this area right here, you have the mitochondrial matrix. And then this phospholipid bilayer is the inner mitochondrial membrane. And then what you have embedded are these transmembrane proteins that will act like pumps. So this is a hydrogen pump, hydrogen pump, hydrogen pump. And then also you have the enzyme ATP synthase embedded in there as well. And then over here, this area is the intermembrane space. So basically that is the space between the inner membrane and then the outer membrane. Inter means between. Okay, so this is the final step. You have gathered as much NADH and FADH2 that are carrying electrons and protons. And then what they do is that they come to this last stage. You have your NADH first. It'll drop off those electrons. And then it'll drop off the hydrogen ion. And then it'll go back to being NAD+. And then this NAD+, it'll go and pick up some more electrons and pick up some more protons so that it can carry out cellular respiration again. So here, these electrons that it dropped off, it'll go and energize this first protein. And then this protein is just an electron carrier protein that acts like a pump that will pump hydrogen ions from the mitochondrial matrix into the intermembrane space. So hydrogen ions will go from the matrix into the intermembrane space. And then so those electrons, after it's finished with this protein, it'll go to this mobile electron carrier. So this is not a pump, so it won't pump any hydrogen ions. So after that, it'll go to this third protein that is an electron carrier. And then these electrons, what they do is that they'll energize this protein to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. And then now these electrons, it'll go to this mobile electron carrier. And then finally to this last protein, number 4 protein, that will pump hydrogen ions from the matrix into the intermembrane space. And then now these electrons, okay, it'll combine with oxygen and protons to make water. But here you have this high concentration of hydrogen ions, okay? So that's from your NADH. It starts from protein 1. Your FADH2 will be a little bit different. It'll actually start at protein 2, which is not a pump. It is not a pump because it does not span the membrane like these proteins. And then so the electrons will go here, and then electrons will go here, and then electrons will go here, where it will act as a pump and move protons from the matrix into the intermembrane space, so some more hydrogen ions. And then these electrons will go to this mobile electron carrier, and then finally to this 4th protein that will pump some more hydrogen ions from the matrix into that intermembrane space. So you added more hydrogen ions, you've created a higher concentration of hydrogen ions, and then this will represent potential energy that will go through this enzyme called ATP synthase. So these hydrogen ions will go from the intermembrane space back into the mitochondrial matrix through this enzyme, through this ATP synthase. And what it does is that it'll power this enzyme, it'll activate this enzyme to turn ADP into ATP. So now it'll make a lot of ATP because of all of these hydrogen ions activating this enzyme to make ATP. And then that part is called chemiosmosis. And then these electrons going through this series of proteins, that's your electron transport chain. these electrons are activating or energizing these protein pumps to move hydrogen ions from the matrix into that intermembrane space. And then it builds up this concentration gradient that will be used in addition to ATP synthase to make a lot of ATP, So now you ask the question, well, what about all those electrons that, finally went to this fourth protein? Where do they go? And then these hydrogen ions that finally came back into the matrix, well, they will combine with oxygen. The hydrogen ions will combine with oxygen. The electrons will combine with oxygen. And then you'll form water. So we say that oxygen in cellular respiration is the final electron acceptor. Okay, so please go through this picture, take it step by step, follow the arrows. Okay, you do need to know that per molecule of NADH, you will make 3 ATP. So for one molecule of NADH, you'll make 3 ATP. So what if there was like a math question? What if you had 3 NADHs? How many molecules of ATP can you make from 3 molecules of NADH? Well, 3 times 3 is 9. So you can make 9 ATPs from 3 NADH. And then also you need to know that per molecule of FADH2, you can make 2 ATPs. So what if we had 3 molecules of FADH2? How many ATPs can you make from 3 molecules of FADH2? Well, you can make 6. 6. Okay. So what if I ask you a question? Carbon monoxide and cyanide are known to, is a poison that can block these electron carriers. Specifically, it can block complex 4 in the electron transport chain. So if you block this, what would happen to you, to your cell? What's going to happen is that you won't be able to make as much hydrogen ions, the concentration, and you won't be able to make as much ATP. In fact, you'll cease to make any ATP, and then you'll eventually die. So that's how the poisoning will work, is that it'll block this complex here, and then if you block hydrogen ions from entering the matrix into the intermembrane space, you won't have it to activate your ATP synthase enzyme to make the necessary ATP to sustain your cells. And then also in bacteria, there's medications that will block their enzyme, their ATP synthase. So what happens when we take antibiotics that specifically blocks their enzyme, their bacterial ATP synthase? They will die. The bacteria dies and then the infection goes away. Because if this enzyme is blocked, all this hydrogen ions, it can't go from the intermembrane space into the mitochondrial matrix. And then you can't make the necessary ATP for that bacteria to keep on living, to keep on causing pathogenesis or disease. So in your book, please take a look at this picture. Your textbook does a great job of explaining it. So that is in the case if you do have oxygen, if you are breathing normally, if you're not exercising. But there's something that's called fermentation, where you don't have any oxygen. So fermentation, please associate it with anaerobic. So in these anaerobic fermentation stages, you actually don't make much ATP. So for us, this anaerobic state, hopefully it'll just be a temporary state, and then we can breathe in oxygen where we can go into the mitochondria and make a lot of ATP. So there's two types of fermentation. You have lactic acid fermentation and you have alcoholic fermentation. And then these fermentation steps, they occur in the cytoplasm. Because you don't have any oxygen, because you are in an anaerobic state. And then this fermentation is necessary so that we can recycle, so that we can make our NAD+, so to be ready for when we do have oxygen. So take, for example, we actually undergo lactic acid fermentation. The common step in fermentation and cellular respiration is glycolysis. Okay, so glycolysis, remember, occurs in the cytoplasm, is anaerobic. When you are undergoing glycolysis, you make ATP, you make 2 ATP, a net of 2 ATP. And then you turn 2 NAD plus into 2 NADH that will carry each 2 electrons and 1 proton. Okay, and in addition, you've made 2 pyruvates. So here, if we don't have any oxygen, then we can't go into the mitochondria and go into that last step to turn this NADH back into NAD+. So this fermentation is a way that will allow our cells to temporarily turn these NADHs back into NAD+. So that when we do have oxygen, we can go through glycolysis, we can go through formation of acetyl coenzyme A, we can go through Krebs cycle, we can go through the electron transport chain and chemiosmosis to make a lot of ATP. So here, this is that same picture of lactic acid fermentation. So this is what happens in our cells, our animal muscle cells, when we're exercising. Okay, so when you're exercising, you are using up more ATP than you can produce. You are not breathing, you're using up all the oxygen that's needed to make the ATP for you to keep exercising. So you are temporarily in an anaerobic state. So you undergo lactic acid fermentation, which just means that you're undergoing glycolysis like normal. You're making your 2 net ATP, 2 NADH, 2 pyruvates, and then you have your lactic acid fermentation that will allow this NADH to go back to being NAD+, so that when you do have oxygen, this NAD+ is ready to go through cellular respiration to make a lot of ATP, because you need oxygen to go into the mitochondria. So here in lactic acid fermentation, which happens in our cells and some bacteria, your products are 2 net ATP and 2 lactic acids, or 2 lactates. The other type of fermentation is called alcoholic fermentation, and then this happens in yeast cells and some bacterial cells. And then here, fermentation, of course, means anaerobic, no oxygen. And then this happens in the cytoplasm. And then here you go through glycolysis again. You make your 2 pyruvates and then your 2 NADHs. So those 2 NADHs will revert back to being NAD+. So when the yeast has oxygen again, it can go and pick up some more electrons and pick up some more protons to undergo cellular respiration. So here, the products of alcoholic fermentation are 2 net ATP, 2 carbon dioxides, and 2 ethanol molecules. So ethanol is just an alcohol. And then this carbon dioxide, well, is used to make products that we use. It's used to make bread. It's what makes the bread rise, that loaf of bread rise. It's used to make cheese as well. And then also is used to make alcohols that people will consume, beer, wine. Those will undergo fermentation. So the 2 ATPs that you've made from both fermentation, lactic acid fermentation and alcoholic fermentation, is made by glycolysis. So here in this lactic acid fermentation, since you're only making 2 ATP per fermentation, you can't sustain fermentation for that long. You need that oxygen to undergo cellular respiration so you can make a lot of ATP. So that's why we say that this lactic acid fermentation is only temporary. And then that is cellular respiration. So I hope you have a better understanding of this process. It's important that you try to find other videos to help you. Khan Academy is really good. Crash Course Biology is really good. And then you can just watch videos that has moving parts to help you better understand this process. There's another worksheet that I find very helpful. It's a flow chart, and then each bubble, there's specific things that you can put in each bubble. Each bubble, you can put more than one type of molecule, okay? So use this and try to be as specific as possible. And then, of course, I have uploaded the answers to this flow chart in the worksheets assessment